Something changed in the human population around the year 1900. Not in the way most people think about historical change. Not through war, or politics, or the rise and fall of governments. This change was quieter than that. It was biological, and in many ways, that makes it harder to explain, and harder to ignore. When we talk about biological change in a population, we are talking about something that happens slowly, over many generations, the kind of change that takes centuries to become visible, and thousands of years to become significant. That is how biology is supposed to work. Gradual, measurable, traceable. But something about the blood type picture of the human population around the start of the 20th century does not quite follow that pattern. And the more you look at it, the more difficult it becomes to explain using the standard framework we have been given. Blood types are something most people only think about when they need a transfusion, or when they are filling out a medical form, or perhaps when they read one of those articles about personality traits linked to your blood group. For most people, a blood type is just a letter and a symbol. AB, or O, positive or negative. It seems simple. It seems fixed. And in one sense it is. Your individual blood type does not change throughout your life. But the distribution of blood types across an entire population, the percentage of people who carry each type, that is something that can shift over time. And it is in that shifting distribution where something unusual appears to be hiding. Researchers who study population genetics spend their careers tracking exactly these kinds of shifts. They look at how genetic traits move through populations over time, how they respond to environmental pressure, how they change after migration events, and how they reflect the deep history of a group of people. Blood type distribution is one of the tools they use. It is not the only tool, but it is a valuable one. And several of these researchers have noted, quietly, that the global picture of blood type distribution does not tell a perfectly clean story. There are patterns in the data that don't fully match what the standard historical narrative would predict. And the explanation for why those patterns exist is not as settled as most people would assume. Here is where the first layer of difficulty appears. The ABO blood type system, the framework that allows us to categorize human blood into these four major groups, was only formally identified and described in 1901. The Austrian scientist Karl Landsteiner published his findings that year, and his work eventually earned him the Nobel Prize in Physiology or Medicine in 1930. His discovery was genuinely important. Before it, blood transfusions were dangerous and often fatal, because no one understood why some transfusions worked and others caused the body to react violently. Landsteiner's work changed that. It saved lives, and it gave science a new tool for understanding human biology. But here is the problem that this timeline creates. If the ABO blood type system was only identified and named in 1901, then we have no systematic, large-scale blood type data from before that point. There are no surveys from 1850. There are no population-wide blood type records from 1800 or 1750 or 1700. The science simply did not exist in a form that would allow that kind of data to be collected. And so, when researchers want to understand what blood type distribution looked like before the 20th century, they are working with an almost completely empty record. That gap, that blank period in the biological history of humanity, begins precisely at the moment when, according to some historical researchers, major and poorly understood disruptions were taking place across large parts of the globe. This is where it becomes slightly unclear, because the absence of data is not the same as evidence that nothing unusual happened. The absence of data simply means we cannot see clearly. And when you cannot see clearly, you are working on assumption. The assumption built into mainstream science is that blood type distributions before 1900 looked roughly similar to what we measured in the early decades of the 20th century, that the distribution was stable, gradual, and continuous. But that assumption has never been fully tested. It cannot be fully tested because the data required to test it does not exist in any reliable form. Think about what this means for a moment. We are told that the current global blood type map, the distribution of type O, type A, type B, and type AB across different regions and populations, reflects thousands of years of gradual human evolution and migration. And that may be true, but the actual measurements that created this map, were taken in a window of time beginning around 1910 and expanding through the following decades. The map is not a picture of ancient humanity. It is a picture of the population that existed after 1900, and if something happened to that population, or to a previous population, in the decades or centuries just before those measurements were taken, the map would reflect the result of that event, not the history before it. This raises a question that is simple to state, but surprisingly difficult to answer. What exactly did blood type distribution look like before 1900? Not as a theoretical projection, not as a model built from modern measurements, but as a real, documented, measurable fact. And the honest answer is that we do not know. We have fragments. We have ancient DNA studies that attempt to extract blood type information from skeletal remains. We have some indirect evidence from historical medical texts that mention blood characteristics, but we do not have anything close to a complete picture. The record before 1900 is thin, and in certain regions of the world, it is almost entirely absent. At first, this seems like a normal limitation of science. After all, we cannot expect detailed biological records from centuries when the relevant science had not yet been developed. That is a reasonable point. But the problem is that this limitation applies unequally across different regions of the world. In some areas, ancient DNA research has filled in parts of the picture. In others, the evidence is consistently harder to find, harder to interpret, or consistently producing results that do not match what the standard model predicts. And it is in those specific regions, the regions where the evidence is most confusing, that the broader historical record is also most incomplete, most contradicted, and most difficult to verify through independent sources. This is where the question starts to feel like something more than a simple gap in the scientific record, because gaps in knowledge are normal, but gaps that consistently appear in the same places, at the same time periods, and across multiple independent lines of evidence. That is a different kind of pattern, and this particular pattern keeps pointing back toward the same general period, the 17th, 18th, and 19th centuries. The period immediately before the blood type data begins to exist. The period during which major population disruptions, poorly documented events, and large-scale historical revisions appear to cluster in the historical record of several regions simultaneously. Some researchers have begun to ask whether the blood type gap and the historical record gap are connected, whether the reason we cannot see clearly into the pre-1900 biological record is the same reason we cannot see clearly into the pre-1900 historical record in certain parts of the world. This is not a claim. It is a question, and it is a question that the standard framework does not have a fully satisfying answer for. The official response is that both gaps are simply the result of limited technology and incomplete record keeping from earlier periods. And that is a reasonable response, but it does not explain why some regions show these gaps far more severely than others. It does not explain why the regions with the deepest historical gaps also tend to be the regions where the earliest blood type studies produced results that researchers found unexpected. And then there is the deeper question underneath all of this. If a population had existed, a large, widespread population with a distinct biological profile, including a distinct blood type distribution, and if that population had been significantly disrupted or replaced over the course of 1 or 2 centuries, what evidence would remain? The answer depends on how complete the disruption was. If the replacement was gradual and partial, you might expect to see traces of the original distribution blending into the new one, a kind of biological echo. If the replacement was more complete, those traces might be very faint. And if the replacement coincided with a period of limited scientific measurement and sparse historical documentation, you might not be able to see the echo at all. You would only see the population that came after, and you would have no reliable way of knowing how different it was from the population that came before. This is where the investigation begins to feel genuinely open-ended, because the tools we have for answering these questions are improving. Ancient DNA technology has advanced dramatically in the last 2 decades. Researchers can now extract and analyze genetic material from skeletal remains that are hundreds or even thousands of years old. And those studies are beginning to fill in parts of the biological history that were previously invisible. But they are also producing results that sometimes complicate the standard model rather than confirming it. Blood type distributions in ancient remains from certain regions do not always match what the standard model would predict. Migration events reconstructed from genetic data do not always align neatly with the migration events described in the historical record. And the regions where these mismatches appear most often are not random. So we are left with a situation that is difficult to resolve cleanly. There was a period, roughly the century and a half before 1900, during which major historical disruptions occurred across multiple regions of the world simultaneously. During that same period, we have almost no systematic biological data about the human populations affected by those disruptions. And when we try to reconstruct what those populations looked like, using ancient DNA, using genetic modeling, using the earliest available blood type studies, the picture we get is not entirely consistent. It is not entirely inconsistent either, but it is uncertain enough that the question deserves to be asked more carefully than it usually is. What exactly was the human blood type picture before 1900? Why is the record so thin in certain regions and so confusing in others? Why does the absence of data align so precisely with the period that would be most relevant to understanding whether something significant changed? These are not questions with easy answers, and that, perhaps, is precisely why they are rarely asked with the seriousness they seem to deserve. The gap in the biological record is real. The gap in the historical record is real, and the overlap between those two gaps is not something that can be dismissed without a more complete explanation than currently exists. Something about this period, and something about the populations that lived through it, remains genuinely unclear, and the further you follow that thread, the more questions appear, not fewer. To understand why the blood type distribution question is so difficult to resolve, it helps to start with what the official explanation actually says, because the official explanation is not vague. It is detailed, structured, and built on decades of research. It has the weight of mainstream biology behind it, and for most people who encounter it, it is entirely convincing. The problems only begin to appear when you move past the surface of that explanation and start looking at the specific details underneath it. The standard scientific position is that the ABO blood type system did not appear suddenly. It evolved over an enormous stretch of time, not centuries, not thousands of years, but millions of years. The genetic markers that determine whether a person is type A, type B, type AB, or type O are ancient. They predate modern humans entirely. Primates other than humans also carry versions of these blood type markers, which scientists point to as evidence that the ABO system has roots deep in evolutionary history. This is one of the reasons the official explanation feels so solid. It is not describing something that happened recently. It is describing a process that unfolded across geological time. According to this framework, type O is the oldest of the four major blood types. The reasoning is that type O results from the absence of certain antigens, the surface proteins that distinguish blood types from one another. In genetic terms, type O represents a simpler version of the blood type gene. And in evolutionary biology, simplicity often indicates age. Type O is thought to have been the dominant blood type in early human populations, carried by the ancestors who first moved out of Africa and spread across the planet. This is why, even today, type O remains the most common blood type globally, and why certain isolated or indigenous populations, particularly in the Americas, show very high frequencies of type O, sometimes approaching 100%. Type A is considered the next development in this evolutionary sequence. It emerged through genetic mutation, producing a new antigen on the surface of red blood cells. Over time, as populations carrying type A genes grew and migrated, the trait spread into new regions. Western Europe became a stronghold for type A over long periods of population settlement and isolation. The explanation for why type A became dominant in Europe and not elsewhere involves a combination of factors: the founding populations that first settled those regions, the specific disease pressures those populations faced, and the degree of genetic isolation that kept the trait concentrated rather than diluting it through mixing with outside populations. Type B followed a different path. It developed independently, not as a continuation of the type A line, but as a separate mutation producing a different antigen. The geographic stronghold of type B is Central Asia and the Indian subcontinent. Researchers link this distribution to the populations that developed and expanded across the Eurasian steppe over thousands of years. As those populations migrated, they carried the type B gene with them. The current map of type B frequency, with its concentration in Central and South Asia and its gradual decline toward the West and East, is presented as a reflection of those ancient migration routes. It is a coherent picture. It follows the logic of population genetics as it is currently understood. Type AB is the most recent of the four major types. It can only exist in a person whose parents carry both the type A gene and the type B gene. This means it could not have appeared in populations before type A, and type B came into contact with each other through migration and mixing. AB is relatively rare globally, and its distribution reflects the areas where type A and type B populations historically overlapped. This rarity and its dependent nature, the fact that it requires both other types to exist first, makes it a useful marker for researchers trying to trace population mixing events. Where type AB appears in significant numbers, it suggests a history of contact between populations that previously carried different dominant blood types. This framework is internally consistent. Each blood type has an origin story. Each geographic distribution has an explanation tied to migration, isolation, or mixing. And the overall picture, with type O concentrated in the Americas and sub-Saharan Africa, type A dominant in Western Europe and parts of East Asia, type B concentrated in Central and South Asia, and type AB appearing in the zones of overlap, makes logical sense within the model. If you accept the model, the distribution is not surprising. It is almost exactly what you would expect, given what we know about ancient human migration patterns. Blood type research moved quickly after Landsteiner's discovery in 1901. Within the first decade, other researchers had already extended the work. In 1902, Alfred von De Castello and Adriano Sturley identified the AB blood type, completing the four-group system. By the 1910s, researchers were already conducting blood type surveys in military populations, hospital patients, and eventually general civilian groups across multiple countries. The first large-scale geographic surveys began appearing in the 1920s, and by mid-century, researchers had collected blood type data from populations on every inhabited continent. This data became the foundation of the global blood type map that is still referenced today. The speed of this research expansion is worth noting. Within roughly 50 years of the initial discovery, scientists had built a comprehensive picture of global blood type distribution and constructed a detailed evolutionary explanation for why that distribution existed. That is a relatively short period in which to establish something that is now treated as settled science. And that speed raises a quiet question, not about whether the researchers were competent, because clearly they were, but about whether 50 years is long enough to be certain that the picture captured is an accurate reflection of the deeper history, rather than a snapshot of a specific moment that may not represent the long-term pattern. At first, this concern seems manageable. After all, science builds on earlier work. The blood type studies of the 1920s and 1930s were refined by later studies. New techniques were developed. Sample sizes increased. Geographic coverage improved. And the results were broadly consistent across different studies conducted in different countries at different times. That consistency is genuinely reassuring. It suggests the measurements are reliable. But consistency in measurement is not the same as completeness in understanding. You can consistently and accurately measure the population that currently exists without that telling you anything reliable about populations that existed before the measurement period began. And this is where the explanation starts to show its first signs of strain, because the measurements that built the global blood type map were all taken after 1901, in most cases after 1910. In many regions of the world, the reliable large-scale data does not begin until the 1920s or even later. This means the foundation of the official explanation, the actual data, is a picture of post-1900 populations. It is a picture taken after whatever happened in the 18th and 19th centuries had already happened. If those centuries contained events that significantly altered the biological composition of certain populations, the data collected afterward would reflect the altered population, and the evolutionary explanation built around that data would be explaining the distribution of the new population, not the original one. This does not mean the evolutionary explanation is wrong. The broad framework, ancient origins, gradual mutation, migration-driven distribution, is almost certainly correct in its general outlines. Human blood types did evolve over long periods. The geographic patterns do reflect ancient migration events. None of that is seriously in doubt at the level of broad principle, but the specific percentages, the specific regional frequencies, the fine-grained details of who carries what type in what concentration in which part of the world, those details are built on data from a very specific and narrow window of time, and the assumption that this window accurately represents the previous 10,000 years is an assumption, not a demonstrated fact. The early data itself contains details that are occasionally difficult to explain cleanly. When researchers in the 1920s conducted blood type surveys in different parts of the world, they sometimes found distributions that did not perfectly match what the theoretical model predicted. Some of these discrepancies were small and were attributed to sampling limitations. Others were larger and prompted adjustments to the theoretical framework. But not every discrepancy was fully resolved. Some were noted, published, and then gradually absorbed into a model that moved past them without providing a complete explanation. The early literature on blood type geography contains these moments, findings that produced slight confusion, that generated competing interpretations, that were eventually simplified in later textbooks into a cleaner narrative than the original data actually supported. This is a normal part of how science works. Early data is messy. Models are adjusted as more information arrives. Discrepancies get resolved over time, and in most cases, the result is a progressively more accurate picture. But the question worth sitting with is whether all of the discrepancies in the early blood type data were actually resolved, or whether some of them were simply set aside as the field moved forward. Because if certain anomalies in the foundational data were not fully explained, just absorbed into a framework that made them less visible, then the official explanation may be more complete-looking than it actually is. The official explanation is logical. It is the product of genuine scientific work conducted by researchers who were doing their best with the tools and data available to them. It answers most of the questions it is asked. But answering most questions is not the same as answering all of them. And the questions it does not fully answer tend to cluster around the same period, the same regions, and the same type of evidence: early data, sparse records, and populations that experienced major disruptions in the centuries immediately before the measurement period began. That clustering is not conclusive, but it is not nothing either, and it is worth following carefully, because the places where an official explanation is least comfortable are usually the places where the most interesting questions live. The first problem with the official explanation is not complicated. It does not require advanced knowledge of genetics or population biology to understand. It is actually quite straightforward once you look at it directly, and that simplicity is part of what makes it worth paying attention to. Simple problems that sit inside large, complex explanations are often the ones that are hardest to see, not because they are hidden, but because the complexity around them makes people assume the problem has already been handled somewhere inside the framework. In this case, it may not have been. The problem is this: almost all of the large-scale blood type population data that forms the foundation of the global blood type map was collected after 1910. The majority of it was collected after 1920, and a significant portion of the most detailed, most geographically comprehensive data was not collected until after 1945. This is not a fringe observation. It is simply what the historical record of blood type research shows. The science was new. The tools were new. And large-scale population surveys take time and resources to conduct. So it is entirely understandable that the data came when it did. But understandable does not mean irrelevant. The timing of when this data was collected has real implications for what it can and cannot tell us. When researchers talk about the global blood type map, the distribution of type O, type A, type B, and type AB across different regions and populations, they are referring to a picture built from measurements taken in the 20th century. In most cases, from measurements taken in the middle decades of the 20th century. That picture is presented as a reflection of deep human history, as a biological record of ancient migration and evolution stretching back thousands of years. And in a broad, general sense, it may well be. But there's a difference, a meaningful, important difference, between a picture of ancient populations and a picture of the populations that survived into the 20th century. And that difference is precisely what makes the first doubt worth examining carefully. Populations change. They always have. Migration, disease, famine, conflict, and displacement have been reshaping human populations for as long as humans have existed. When a population experiences a severe disruption, a catastrophic epidemic, a forced migration, a violent conquest, the genetic composition of that population can change significantly within just a few generations. Blood type frequencies can shift. The proportion of people carrying each type can move dramatically if the disruption affects one group more severely than another. And if that disruption happens in the generations just before systematic blood type measurement begins, the measurements will capture the post-disruption population, not the pre-disruption one. There will be no record of what was lost, no comparison point, just the new picture, presented as if it represents the long, continuous history of that region. This is where the doubt begins to take shape, because the century and a half immediately before large-scale blood type measurement began, roughly 1750 to 1900, was not a stable, quiet period in human history. It was marked by some of the most significant population disruptions in recorded history. Entire regions experienced near-total population collapse and replacement. Large-scale famine events in multiple parts of the world reduced populations by millions within short periods. The Atlantic slave trade forcibly displaced and mixed populations on a massive scale. Colonial expansion destroyed or severely disrupted indigenous populations across the Americas, Africa, Asia, and the Pacific, and in Central and Eastern Eurasia, the regions most associated with the Tartarian hypothesis. Enormous political and military transformations reorganized populations in ways that historians are still working to fully understand. All of this happened before the blood type measurements were taken, and yet the measurements are used to make claims about what ancient populations looked like thousands of years ago. At first, this seems like a manageable limitation. Scientists account for historical disruptions in their models. They adjust for known migration events. They use genetic data to reconstruct population histories. But the adjustment process depends on knowing which disruptions occurred, how severe they were, and which populations were affected. If certain disruptions are poorly documented, or if the scale of a population replacement event is not fully recognized by the historical record, the adjustment cannot account for it. The model will simply treat the post-disruption population as continuous with the ancient one, and the blood type distribution of the replacement population will be mapped onto a history it may not actually belong to. Ancient DNA research was supposed to help resolve this problem, and in many ways it has. The ability to extract and analyze genetic material from skeletal remains that are hundreds or thousands of years old has opened a window into population history that was completely closed just a few decades ago. Researchers can now identify blood type markers in ancient remains and compare them to the distribution seen in modern populations from the same geographic region. This comparison is exactly the kind of check that should help reveal whether modern populations are genuinely continuous with ancient ones, or whether a replacement event occurred at some point in between. But this is where it becomes unclear, because the results of ancient DNA research have not always confirmed the continuity that the standard model assumes. In some regions, the ancient blood type distributions match the modern ones reasonably well. The change from ancient to modern is gradual, traceable, and consistent with the slow process of genetic drift and migration that population genetics describes. That is what you would expect if the population in question is genuinely continuous across time. Gradual evolution leaves gradual fingerprints. The genetic change accumulates slowly, predictably, and in ways that researchers can follow step by step through successive periods. But in other regions, the picture is different. Some ancient DNA studies examining European remains from pre-medieval periods have found type A frequencies that do not match what current European populations show. Other studies have found type B appearing in ancient populations from regions where type B is now quite rare, regions where the standard model would predict it was never common. And in several documented cases, researchers have published findings showing that the blood type distribution in ancient skeletal remains from a particular area simply does not match the modern population living in that same geographic area today. The ancient people and the modern people, in terms of blood type distribution, look like different populations. At first, each of these individual findings can be explained. Migration could account for it. A disease event could account for it. A known historical population movement could account for it. And often, these explanations are offered and accepted. But the question worth sitting with is whether the explanations are being applied consistently, because migration and disease events are used to explain discrepancies in both directions. When ancient populations look different from modern ones in ways that don't fit the model, the explanation is migration or disease. But when modern populations look different from ancient ones in ways that do fit the model, the continuity is simply assumed. The same mechanism, population disruption and replacement, is cited as an explanation in one context and ignored as a possibility in another. And that inconsistency is worth noticing. The key distinction is between gradual change and abrupt change. This is not a subtle difference. In genetic terms, it is a clearly detectable one. When a population changes gradually over time, through slow migration, through generations of mixing with neighboring groups, through incremental genetic drift, the signal left in the genetic record is gradual. You can see the change accumulating across successive archaeological periods. The blood type frequencies shift slowly. The haplogroup distributions blend at the edges. The overall picture tells a continuous story with no sudden breaks. That is the kind of fingerprint that gradual evolution leaves. And when researchers find it, the standard model is well equipped to explain it. But abrupt change leaves a different fingerprint entirely. When a population is replaced rapidly, through conquest, through epidemic, through forced displacement on a large scale, the genetic signal does not show a gradual transition. It shows a break, a point in the record where one distribution ends and a different one begins. Sometimes the break is partial. The new population mixes with the remnant of the old one, and the fingerprint shows a sharp shift followed by a period of blending. Sometimes the break is more complete. The old distribution simply stops appearing in the archaeological record, and the new one takes over without an obvious transitional phase. Researchers who study ancient DNA know what these different patterns look like, and some of them have noted, in published research, that certain regions show break-type fingerprints where the standard model predicts gradual-type fingerprints. This matters because it is not just one study, or one region, or one period. The pattern of unexpected results in ancient DNA research, blood type distributions that don't match the modern populations of the same regions, appears across multiple independent studies conducted by different research teams in different parts of the world. Not everywhere, not even in most places, but in enough places and in enough consistent geographic clusters that it becomes difficult to attribute entirely to sampling error or to the general noisiness of early genetic research. At some point, a pattern that repeats across independent data sources stops looking like coincidence and starts looking like a signal. The question is what that signal is pointing toward. Some researchers believe the answer lies in events that the standard historical record does not fully capture. That certain population replacement events, occurring in the 17th, 18th, or 19th centuries, were large enough and complete enough to significantly alter the blood type distribution of the affected regions, but are not represented in the official historical narrative in a way that allows the genetic models to account for them. Others believe the anomalies will eventually be explained by refinements in the ancient DNA methodology itself, that as sample sizes grow and techniques improve, the unexpected findings will resolve into patterns that fit the standard model. Both positions are reasonable. Neither has been definitively proven. What can be said with reasonable confidence is this. The global blood type map is a picture of post-1900 populations. Ancient DNA research shows that in certain regions, those populations carry blood type distributions that do not smoothly connect to the ancient populations of the same areas. The fingerprints left in the genetic record in those regions look less like gradual evolution and more like abrupt replacement. And the period during which that replacement most likely would have occurred, if it occurred, is precisely the period for which we have the least documentation, the least data, and the least ability to independently verify what actually happened. That combination of factors does not produce a conclusion, but it does produce a question that is genuinely difficult to answer using the information currently available. And the difficulty of answering it is not a reason to stop asking. To understand why the blood type question carries the weight it does, it helps to step back from the biology for a moment and look at the historical period surrounding it. Because the science does not exist in isolation. It exists inside a specific window of time. And that window, the late 1800s and the early 1900s, was not a calm or stable period in human history. It was one of the most turbulent and demographically disruptive periods the modern world has ever seen. Understanding the scale of what was happening to human populations during this time is essential for understanding why the blood type data, collected immediately afterward, might not tell the complete story it is assumed to tell. Start with the Americas. By the time the 19th century began, the indigenous populations of North and South America had already been experiencing catastrophic decline for roughly 300 years. The arrival of European colonizers in the late 15th century introduced diseases to which indigenous populations had no immunity. Smallpox, measles, typhus, and other illnesses moved through these communities with devastating speed. Estimates of the pre-contact population of the Americas vary widely, but many historians and demographers believe the indigenous population may have been reduced by 80% or more over the course of 3 centuries. Some regions experienced near-total population collapse. Entire cultures, languages, and genetic lineages were lost. And the populations that survived into the 19th and 20th centuries, both indigenous remnants and the settler populations that replaced them, carried a very different biological profile than the populations that had existed before contact. If you measured blood types in the Americas in 1920, you were measuring the result of 3 centuries of replacement, not the original picture. The situation in Central Asia and Eastern Europe during the same period was different in character, but similar in its implications for population genetics. The great territorial empires that had dominated these regions, the Ottoman Empire, the Russian Empire, the various Central Asian Khanates, were either collapsing, contracting, or being violently reorganized throughout the 19th century. These were not peaceful administrative transitions. They involved large-scale military campaigns, forced resettlements, mass deportations, and in several cases, deliberate efforts to replace one population with another in strategically important territories. The Russian Empire's expansion into Central Asia and the Caucasus during the 19th century, for example, involved the displacement and partial replacement of numerous distinct ethnic and genetic populations. The populations measured by early 20th century blood type researchers in these regions were the populations that existed after these processes had already run their course. The Ottoman Empire's slow dissolution across the 19th and early 20th centuries created its own set of demographic upheavals. Populations that had lived in the same territories for centuries were displaced, migrated, or destroyed as the empire's administrative structures broke down and new national boundaries were drawn. The Balkan Wars of the early 20th century, occurring almost simultaneously with the period when early blood type surveys were being conducted, produced massive population movements across southeastern Europe. Greeks, Turks, Bulgarians, Serbs, and dozens of smaller ethnic groups were displaced in enormous numbers. The populations that blood type researchers measured in the 1910s and 1920s in this region were populations in the middle of, or immediately following, one of the most significant demographic reorganizations in European history. Russia itself was undergoing transformations of extraordinary scale during this period. The emancipation of the serfs in 1861 triggered massive internal migration as tens of millions of people who had been legally bound to specific territories became free to move. Industrialization drew populations from rural areas into rapidly growing urban centers. The famines of the late 19th century, particularly the severe Russian famine of 1891 and 1892, caused enormous mortality concentrated in specific geographic regions and specific demographic groups. Then came the upheavals of the early 20th century, the 1905 Revolution, the First World War, the 1917 Revolution, the subsequent Civil War, and the famines of the early 1920s. Each of these events reshaped the Russian population in ways that were genetically significant. By the time large-scale blood type surveys were conducted in Soviet territories, the population had been through multiple severe disruption events in rapid succession. And here is where it becomes important to connect this historical context to the biological question. Each of these events, the indigenous population collapse in the Americas, the imperial reorganizations in Central Asia, the Ottoman dissolution, the Russian upheavals, was a population genetics event, not just a political or social event. When large numbers of people die in a specific region, when they are replaced by migrants from a different genetic background, when the surviving remnant mixes with an incoming population, the blood type distribution of that region changes, sometimes gradually, sometimes quite sharply. And if those changes happen in the decades just before systematic blood type measurement begins, the measurements will capture the post-change distribution. The original distribution, whatever it was, will be invisible. This is not a speculative concern. It is a documented reality in mainstream population genetics. Researchers have a term for it, a population bottleneck. A bottleneck occurs when a population is dramatically reduced in size by some catastrophic event, and the surviving group becomes the ancestor of all subsequent members of that population. The genetic diversity of the original population is not preserved in the bottleneck survivors. Only the genetic profile of the survivors continues forward. Blood type frequencies can shift dramatically through bottleneck events, because the survivors may not represent the same distribution as the original population. And if the bottleneck is followed quickly by population replacement, incoming groups filling the territory left vacant by the reduced original population, the resulting genetic picture can look very different from anything that existed in that region before the bottleneck occurred. It is within this historical context that the Tartarian theory becomes relevant, not necessarily as a confirmed historical account, but as a pointer toward a set of questions that the standard historical framework does not fully address. The Tartarian theory, which has circulated primarily in alternative research communities, proposes that a large and sophisticated civilization once occupied the territories of Central Eurasia, the vast landmass stretching from Eastern Europe through Siberia and into Central Asia. According to this theory, this civilization was deliberately suppressed and erased from history during the 18th and 19th centuries. Its people were replaced, its records destroyed or rewritten, and its physical infrastructure either demolished or absorbed into the historical narrative of other civilizations. This is a significant claim, and it should be treated with appropriate caution. The evidence cited in support of the Tartarian theory is often ambiguous, and the interpretations offered are frequently more speculative than the presentation suggests. Mainstream historians do not accept the theory in the form it is usually presented. The cartographic evidence, the fact that old European maps labeled vast areas of northern Asia as Tartaria, is acknowledged but explained as a geographic convention rather than evidence of a unified civilization. The architectural anomalies cited by Tartarian researchers are generally attributed to known historical cultures, and the claim that an entire civilization was deliberately erased from history raises obvious questions about how such an erasure would have been coordinated and concealed across multiple competing nations and record-keeping traditions. But here is where the distinction becomes important. Whether or not the Tartarian theory is accurate in its specific claims, the general mechanism it describes is not unusual or impossible. Population replacement is not a fringe concept. It is a well-documented reality in mainstream history. The replacement of indigenous populations in the Americas is taught in schools. The displacement of populations during the Ottoman dissolution is documented in academic history. The deliberate resettlement policies of the Russian Empire, which moved populations around Central Asia to serve strategic purposes, are part of the historical record. These are not controversial claims. They are accepted facts, and they demonstrate that the mechanism the Tartarian theory invokes, large-scale population replacement, is real, documented, and historically significant. The question the Tartarian framework raises, stripped of its more speculative elements, is actually a reasonable one. It is simply this: is it possible that a large-scale population replacement occurred in Central Eurasia during the 17th, 18th, or 19th centuries at a scale that has not been fully acknowledged by mainstream historical scholarship? Not necessarily involving a technologically advanced secret civilization, just a large population, with its own genetic profile that was substantially displaced or destroyed and replaced by a different population, and that this event is either poorly documented, actively understated, or not fully integrated into the genetic and historical models currently used to interpret the region's past. This is not a question that can be answered easily, but it is a question that the historical context of the 19th century makes genuinely worth asking. Because if you look at the scale of the disruptions that occurred across Central Eurasia during this period, and if you consider the degree to which the historical record of this region is fragmentary and contested, and if you then consider that the blood type measurements collected in this region in the early 20th century showed distributions that sometimes surprised researchers, then the combination starts to feel like more than coincidence. It starts to feel like a pattern, and patterns and independent lines of evidence are worth following carefully, even when no single line of evidence is strong enough to support a firm conclusion on its own. What mainstream history acknowledges clearly is this: populations were replaced in many parts of the world during the 18th and 19th centuries. The biological consequence of population replacement is that the blood type distribution of the original population is replaced along with it. The blood type measurements taken in the early 20th century reflect the populations that existed at that moment, not the populations that existed before the disruptions of the preceding 2 centuries. And the gap between those two populations in certain regions may be larger than the standard model currently assumes. How large that gap is, and what it would mean for our understanding of human biological history, is a question that the available evidence cannot yet fully answer. But the context makes clear that the question is not unreasonable, and that may be the most important thing this chapter of the investigation establishes. When you move past the general framework and start looking at specific details in the blood type data, the inconsistencies become harder to dismiss. General patterns can absorb a lot of variation without breaking, but specific inconsistencies, the kind that appear in particular blood types, in particular geographic regions, in particular time periods, are more difficult to explain away. And there are several of them in the blood type record that have been noted by researchers over the years, acknowledged in the literature, and then quietly set aside without fully satisfying resolution. The most specific and most persistent of these inconsistencies involves type B blood, and once you look at it carefully, it is not a small problem. Type B blood today is most heavily concentrated in a specific geographic band. Central Asia is its stronghold. The Indian subcontinent carries elevated frequencies. Parts of East Asia show significant type B presence as well. Moving westward from Central Asia, the frequency drops. It drops steadily through the Middle East and Eastern Europe, and by the time you reach Western Europe, type B is comparatively rare. In some Western European populations, particularly in the far northwest, it drops to very low frequencies indeed. This geographic gradient, high in Central Asia, declining sharply toward the west, is one of the more visually clear patterns on the global blood type map, and the standard explanation for it is straightforward. Type B, in this explanation, originated in or spread through Central Asian populations. As those populations migrated westward over thousands of years, they carried type B with them, but migration diluted the frequency as it mixed with existing populations. Western Europe, less affected by these eastward migrations, retained a lower type B frequency, clean, logical, internally consistent. But this is where it becomes unclear, because ancient DNA research, the tool that should confirm this picture by showing the type B gradient gradually establishing itself across the archaeological record, has not always done so. Several studies examining skeletal remains from pre-medieval Europe have found type B appearing in populations where, according to the standard model, it should be rare or absent. These are populations from periods before the major westward migration events that the standard model uses to explain type B's limited presence in Europe. If type B was rare in Western Europe because westward migration never carried it there in significant numbers, then pre-migration European remains should show very low or absent type B frequencies. In some studies, they do, but in others, they do not. And the inconsistency is not small enough to disappear quietly into sampling error. The problem runs in the other direction as well. Some studies of ancient remains from Central Asian regions, from periods predating the major migration events that supposedly established type B dominance there, have produced distributions that do not cleanly match what the standard model predicts. If type B developed and concentrated in Central Asia over a very long period before migrating outward, you would expect to see it building gradually in the Central Asian record, dominant, stable, consistently high through successive archaeological layers. But in certain studies, the picture is more variable than that. The frequency fluctuates. It appears in some periods at levels that are hard to reconcile with the standard model's timeline of development. And the explanation for these fluctuations, when one is offered, tends to invoke localized migration events or sampling limitations rather than addressing the broader question of whether the type B distribution story is actually as well understood as it appears. This does not mean the standard explanation is wrong. It means the data supporting it is less clean and less complete than the confident presentation of the global blood type map implies. There's a difference between a well-tested model and a well-presented model. And the type B distribution story, on close examination, looks more like the latter than the former. The model fits the broad geographic pattern, but when you zoom in on specific time periods and specific regions, the data does not always align with the story the model tells. That misalignment matters, not because it proves something different happened, but because it means the standard model has not been as rigorously tested against the archaeological record as it would need to be in order to be treated as fully confirmed. Then there's the RH factor. And this is where the inconsistency becomes genuinely difficult to resolve, regardless of which broader framework you are working within. The RH factor is a separate blood classification system from the ABO system. While ABO categorizes blood by the presence or absence of A and B antigens, the RH system categorizes blood by the presence or absence of a protein called the RH antigen, also called the D antigen. If your red blood cells carry this protein, you are Rh positive. If they do not, you are Rh negative. The majority of the global population is Rh positive. Rh negative blood is the minority, but it is not a uniformly distributed minority. It is highly concentrated in specific geographic locations, and that concentration is one of the things that makes it difficult to explain through standard evolutionary mechanisms. Rh negative blood is most common in the Basque population of northern Spain and southern France. The Basques are a linguistically and genetically distinct group whose origins are not fully explained by mainstream population genetics. Their language, Basque or Euskera, is a language isolate. It is unrelated to any other known language in the world. Their genetic profile sets them apart from surrounding European populations in several significant ways, and their Rh negative frequency is the highest of any population group on Earth, reaching levels that are far above the global average. Beyond the Basques, elevated Rh negative frequencies appear in certain parts of the British Isles, in some North African populations, particularly Berber groups, and in scattered pockets across Western Europe. The distribution is not random, but it is also not smooth. It clusters, it concentrates, and that clustering pattern is difficult to explain through the standard model of gradual mutation and spread. The evolutionary problem with Rh negative blood is specific and well recognized. When an Rh negative woman carries an Rh positive fetus, which can easily occur if the father is Rh positive, her immune system can develop antibodies against the Rh antigen on the fetus's blood cells. This condition, called Rh incompatibility, can cause the mother's immune system to attack the red blood cells of the fetus. In a first pregnancy, this typically does not cause severe problems because the immune response develops gradually. But in subsequent pregnancies, if the fetus is again Rh positive, the pre-existing maternal antibodies can cross the placenta and attack the fetal blood cells with much greater force. This can cause a condition called hemolytic disease of the newborn, a serious and potentially fatal condition that, without modern medical intervention, would result in the death of the affected infant. This creates a significant natural selection problem, a trait that causes the death of infants, particularly in pregnancies after the first, should be strongly selected against over evolutionary time. The mothers who carried the Rh negative gene would have lower rates of successfully raising multiple Rh positive children to adulthood. Over generations, this should reduce the frequency of the Rh negative gene in the population. Natural selection is not a perfect filter, but it is a powerful one over long periods. And yet Rh negative blood not only persists in certain populations, It persists at relatively high frequencies. The Basque population, where it is most concentrated, shows Rh-negative rates that selection pressure should have reduced significantly over the thousands of years this population has existed. The fact that it hasn't been reduced, or hasn't been reduced as much as the selective pressure would predict, requires an explanation. At first, several explanations have been proposed. One is that Rh-negative confers some survival advantage that offsets the reproductive disadvantage. Researchers have explored whether Rh-negative individuals show greater resistance to certain parasites or diseases. Some studies have suggested possible connections to resistance against some viral infections, but none of these proposed advantages have been demonstrated clearly enough and consistently enough to fully account for the persistence of Rh-negative at the frequencies seen in Basque and related populations. The evolutionary math doesn't quite work out, and this is where the explanation starts to weaken, not catastrophically, but noticeably. Another proposed explanation is that the Rh-negative trait is simply old enough, and the population carrying it small enough and isolated enough, that genetic drift has preserved it despite the selective disadvantage. Genetic drift is the random fluctuation of gene frequencies in small populations. In a small, isolated group, a trait can persist or even increase in frequency, simply because of chance, regardless of its selective value. The Basque population is and has historically been relatively isolated. Their linguistic and genetic distinctiveness supports this. So drift could theoretically explain some of the persistence. But drift alone is a weak explanation for a trait that persists at such high frequencies over such a long period. Drift usually produces random fluctuation, not stable high frequencies. And the geographic clustering of Rh-negative blood, its concentration in the Basques, with echoes in nearby Atlantic populations, suggests something more structured than pure chance. What makes the Rh factor situation particularly interesting in the context of this broader investigation is the question of origin. Where did Rh-negative blood come from? Every genetic trait has an origin point, a population in which the relevant mutation first appeared. For most blood-type traits, researchers can at least propose a plausible geographic origin based on where the trait is most concentrated and how its frequency diminishes with distance from that origin. For Rh-negative, the geographic concentration in the Basque region suggests an origin somewhere in southwestern Europe or the broader Atlantic fringe. But the Basques themselves are genetically unusual in ways that make their origin story one of the more contested questions in European population genetics. They appear to be a remnant of a very ancient European population, possibly predating the agricultural migrations that reshaped most of Europe's genetic landscape. But the details of their ancestry and the origin of their distinctive traits, including Rh-negative blood, remain genuinely unclear. And this is where the two inconsistencies, the type B distribution problem and the Rh-negative persistence problem, begin to feel like parts of a larger pattern rather than isolated puzzles. Both involve blood-type traits whose geographic distribution and frequency are difficult to fully explain through the standard model of gradual evolution, migration, and natural selection. Both involve regions where the population history is in some way unusual or poorly understood. Type B raises questions about what was happening in Central Asia before the major migration events that supposedly established its distribution. Rh-negative raises questions about what ancient population carried this trait into southwestern Europe, and why it has persisted at levels that selection pressure should have reduced. Neither question has a fully satisfying answer within the current framework. The standard model accommodates both anomalies at a surface level. It offers explanations that are plausible enough to prevent them from being treated as serious challenges. But plausible is not the same as demonstrated. And the more carefully you look at the specific evidence underlying these explanations, the ancient DNA data, the population genetics modeling, the geographic distribution details, the more clearly you can see that the model is working harder than it should need to in order to make the data fit. Data that genuinely fits a model well does not require this much interpretive effort. It tends to confirm the model naturally, with only minor adjustments needed. When the adjustments become frequent, and when they cluster around the same types of anomalies in the same types of regions, it raises the possibility that the model is not fully capturing what actually happened. The data is not fraudulent. The researchers are not incompetent, But the story the data tells is not quite as clean as the standard presentation suggests. And that gap between the clean story and the messy data is where the most important questions in this investigation are beginning to take shape. Among the researchers who have looked closely at blood type distribution history, a specific period keeps appearing in the analysis. It is not a period defined by a single event or a single discovery. It is a window of time, roughly 40 years, spanning from around 1880 to around 1920, that some researchers have begun to describe as a kind of biological threshold moment, a point at which the human population, in certain regions of the world, appears to show characteristics that are difficult to fully account for using the standard models of gradual genetic change. The data from this period is limited. The interpretations are contested, but the pattern is specific enough that it deserves to be examined carefully rather than absorbed quietly into the broader framework without comment. The way researchers identify this threshold is through comparison. On one side, you have the earliest large-scale blood type studies, the surveys conducted in the 1910s and 1920s, which represent the first systematic attempt to measure blood type distribution across significant population samples. These studies are the foundation of everything that came after. They established the baseline numbers that later research built upon and refined. On the other side, you have genetic projection models, computational tools that use known rates of genetic change, migration patterns, and population dynamics to project what blood type distributions should have looked like in the past, given what we measure today. When you run these models backward from the 20th century data, they generate estimates of what the pre-measurement population should have looked like. And in most regions of the world, those estimates align reasonably well with what the early studies actually recorded. But not everywhere. In Eastern Europe and Central Asia, the alignment is weaker. The numbers produced by backward projection models do not match the early study data as cleanly as they do in other regions. The discrepancies are not enormous. They are not the kind of difference that immediately signals an obvious error, but they are consistent. They appear in multiple independent analyses, and they appear specifically in the regions where, according to some researchers, the population history is most likely to contain events that the standard model has not fully integrated. This consistency, the same discrepancy appearing repeatedly in the same geographic cluster, is what elevates the observation from a statistical footnote to something worth investigating more carefully. Eastern Europe and Central Asia are not arbitrary regions. They are precisely the territories that appear on pre-18th century European maps under a specific label. That label is Tartaria, or, in its more expansive form, Grand Tartary. This is not an obscure or marginal cartographic term. It appeared consistently and prominently in some of the most respected geographic and cartographic works produced by European nations during the 16th, 17th, and early 18th centuries. Maps produced in France, Britain, the Netherlands, Germany, and other major European nations all used variations of this label to identify an enormous geographic territory, stretching from the Ural Mountains in the west through Siberia in the north and into Central Asia in the south. The territory labeled as Tartaria on these maps was not a small or peripheral area. It was one of the largest geographic designations on the entire map of the known world. This raises a question that is worth sitting with for a moment. When a geographic label appears consistently across multiple independent cartographic traditions, produced by different nations over a period of more than a century, it is reasonable to assume that label corresponds to something real. Cartographers in the 16th and 17th centuries were not careless. They were working with the best available information, gathered from travelers, traders, diplomats, and explorers. The consistent use of the Tartaria label across these independent sources suggests that the peoples of these regions were recognized by outside observers as forming some kind of identifiable geographic and cultural entity, distinct enough to be given a specific name and a specific territory on maps of the world. And then, over a relatively short period, that label largely disappeared. The transition was not instant, but it was remarkably complete. By the late 18th and early 19th century, the term Tartaria was fading from official cartographic and political usage. By the mid-19th century, it had largely vanished from the maps produced by the same European nations that had used it consistently for generations. The enormous territory it once described was now labeled with different names: Russian Empire, Siberia, Turkestan, various regional designations that carved the formerly unified label into smaller, administratively distinct pieces. The name itself survived in some academic and historical contexts, but as a description of the past, not of a current geographic or cultural reality. Mainstream history offers an explanation for this disappearance. The official account describes the Tartaria designation as a loose, imprecise European label for the diverse nomadic and semi-nomadic peoples of northern and central Asia, the various Turkic and Mongol confederacies that had dominated the Eurasian steppe for centuries. As the Russian Empire expanded eastward and imposed administrative control over these territories during the 18th and 19th centuries, the older, vaguer geographic label was replaced by more specific Russian administrative designations. The peoples previously grouped under the Tartaria label were now subjects of the Russian state, and the cartographic conventions changed to reflect that political reality. This is a coherent explanation. It fits within the standard framework of imperial expansion and administrative reorganization. But this is where it becomes unclear, because the explanation, while coherent, does not fully address the speed and completeness of what occurred. Imperial expansion typically produces a gradual transition in cartographic convention. The old labels persist for a period alongside the new administrative names. Local identities survive in some form even as political control changes. Linguistic and cultural traditions are not erased overnight, and in most cases of imperial expansion in this era, the peoples absorbed into the expanding empire left significant traces in the historical record: their languages, their religious practices, their oral traditions, their physical infrastructure. The degree to which all of these traces would need to have disappeared, and disappeared so thoroughly that even the cartographic label became almost entirely obsolete within a generation or two, is unusual. Not impossible, but unusual enough to invite closer examination. Some researchers have pointed to this speed as one of the more difficult aspects of the standard explanation to accept at face value. They note that the disappearance of the Tartaria designation was not just cartographic. The peoples, cultures, and histories associated with this label became, within a relatively short period, extraordinarily difficult to reconstruct from surviving sources. The documents that would normally provide the most detailed picture of a people's history, administrative records, religious texts, census information, architectural records, are fragmentary, contested, or absent for this region during the critical transition period. What survives tends to be European accounts of the region from the outside, observations by travelers and diplomats, rather than the internal documentary record that would normally accumulate within a functioning civilization or settled population. Now consider what this means in combination with the blood type data. If the population that once occupied the territories labeled Tartaria was substantially replaced during the 18th or 19th century, not through gradual assimilation, but through more rapid displacement, then the blood type distribution measured in these regions in the early 20th century would not reflect that original population. It would reflect the replacement population. The early blood type studies conducted in Eastern Europe and Central Asia in the 1910s and 1920s would be measuring whoever was there after the transition, not whoever was there before it. And the genetic projection models that used that early 20th century data as their baseline would project backward from the wrong starting point. They would generate estimates of what the ancient population should have looked like, but those estimates would be anchored to a post-replacement baseline. The ancient population, whatever its blood type distribution was, would be invisible in the analysis. This is the crux of what makes this period so difficult to assess. It is not that the blood type data is wrong. It is that the data may be accurate for the population it measured, while that population may not be continuous with the population that occupied the same territory in the preceding centuries. If the replacement was significant enough, and if it occurred entirely within the gap before systematic measurement began, there would be almost no way to detect it from the 20th century records alone. The records would simply show the current population, projected backward through genetic models, with nothing to flag the discontinuity. The discontinuity would only become visible if you had independent evidence, from ancient DNA, from pre-replacement historical sources, or from the kind of cartographic and cultural anomalies that some researchers have been collecting, that the pre-replacement population looked different from what the models assume. And some researchers believe they are beginning to find exactly that kind of independent evidence. The ancient DNA studies that have produced unexpected blood type distributions in Central Asian and Eastern European remains. The cartographic record showing a large, consistently labeled territory that was then rapidly and thoroughly erased from official usage. The documentary gaps in the internal records of the region during the critical transition period. The blood type projection discrepancies that appear specifically in the territories formerly labeled as Tartaria. None of these lines of evidence is individually conclusive. Each has alternative explanations available, but the fact that they point in the same direction, toward the same region, the same time period, and the same general type of event, is difficult to dismiss as coincidence without a more complete explanation than currently exists. What the standard explanation struggles most with is the combination. It can explain the cartographic change. It can explain the documentary gaps. It can explain individual blood type discrepancies. But explaining all of these things simultaneously, with a single coherent account that doesn't require any unusual or unacknowledged events, is considerably more difficult. And when an explanation requires that multiple independent anomalies each have their own separate, unrelated cause, rather than a single underlying event that could account for all of them, the explanation becomes progressively less economical. Simple events tend to leave simple patterns. Complex, multidirectional patterns tend to reflect something more significant at their source, whether that source is a population replacement, a deliberate historical erasure, a poorly documented collapse, or simply an accumulation of smaller events that the historical record has not yet fully integrated. That question remains genuinely open. The blood type data from the 1910s and 1920s reflects a specific population in a specific moment. The population it measured may or may not be the same population that occupied that territory 2 or 3 centuries earlier. The cartographic record suggests there was once something significant enough in that region to warrant a consistent geographic label across multiple independent traditions. And then that label, and much of what it once described, became remarkably difficult to find. The biological threshold that some researchers identify in this period may be a reflection of exactly that process. Whatever that process actually was, there is a point in any investigation where individual observations stop being individual, where separate pieces of information, each with its own possible explanation, begin to arrange themselves into something more structured. That point is not a conclusion. It is a pattern, and patterns are significant not because they prove a specific answer, but because they indicate that the random noise of coincidence has given way to something more organized, something that the current framework is not fully explaining. In this investigation, that point has been reached, and the pattern that is emerging deserves to be examined carefully, because it is not coming from one source. It is coming from several independent sources simultaneously, and they are all pointing in the same direction. The first line of evidence is ancient DNA research. Over the past 2 decades, this field has transformed what scientists can know about past populations. The ability to extract genetic material from skeletal remains and analyze it with modern sequencing technology has opened an entirely new window into human history. And what researchers have found through this window has not always matched what they expected. Blood type distributions recovered from ancient remains have, in numerous published studies, produced results that surprised the researchers who conducted them. Not occasionally, not in isolated cases, but consistently enough that the pattern of surprise has itself become a recognized feature of the field. Experienced researchers in ancient DNA now approach new studies with the explicit understanding that the results may not conform to what modern population distributions would predict. That expectation of nonconformity is itself a significant signal. When a scientific field reaches the point where unexpected results are expected, it usually means the underlying model needs revision. What makes this pattern more significant is that the surprises are not random. Random surprises would appear scattered across different regions and different time periods without any clear clustering. Some ancient populations in random parts of the world would show unexpected blood type distributions for no discernible reason that would suggest measurement noise or methodological limitations. problems that would eventually resolve as techniques improved. But that is not what the data shows. The unexpected findings tend to cluster. They appear more frequently in certain geographic regions than others, and they appear more frequently in certain time periods than others. That clustering, geographic and temporal, is what transforms a collection of anomalies into a pattern. It suggests the surprises share a common underlying cause, localized in space and time, rather than being the product of random error. The second line of evidence concerns the geographic overlap between the regions producing the strongest anomalies in ancient DNA research and the regions that experienced the most severe population disruptions between roughly 1600 and 1900. This overlap is not perfect. It is not the kind of one-to-one correspondence that would constitute direct proof, but it is consistent enough to be worth noting carefully. The regions where ancient blood type distributions most frequently diverge from modern ones, Eastern Europe, Central Asia, parts of Northern Asia, are the same regions that experienced the most significant population disruptions during the 3 centuries immediately preceding the start of systematic blood type measurement. This could be coincidence, but coincidence becomes a less and less satisfying explanation the more consistently the same 2 variables appear together across different datasets and different analytical approaches. Population disruptions of the kind that occurred in these regions between 1600 and 1900 are exactly the kind of events that would alter blood type distributions. This is not speculative. It is a straightforward application of population genetics principles. When a population experiences a severe bottleneck, a dramatic reduction in size due to disease, famine, conflict, or displacement, the surviving group carries only a fraction of the original genetic diversity. The blood type frequencies in the surviving group may differ significantly from those in the original population, simply because the survivors are a non-representative sample of the people who came before them. And if the bottleneck is followed by replacement, a new population moving into the territory vacated or partially vacated by the disrupted original, the genetic composition of the region can shift substantially within just a few generations. The blood type data collected afterward will reflect this new composition, not the original one. The third line of evidence is the historical record itself, and this is where the pattern becomes particularly difficult to explain through the standard framework. The official historical documentation for the regions in question, Eastern Europe, Central Asia, the territories formerly labeled as Tartaria on European maps, contains an unusually high concentration of gaps, contradictions, and unresolved revisions for the period between 1600 and 1900. Documents that should exist, based on what we know about the administrative and cultural practices of the populations that inhabited these regions, are frequently described as lost, destroyed by fire, destroyed in conflict, removed to inaccessible archives, or simply absent without explanation. The accounts of what happened in these regions during this period are often reconstructed from secondary sources: the observations of outside travelers, the records of neighboring empires, documents written after the fact by administrators of the powers that eventually took control of these territories. At first, this might seem like a normal feature of historical scholarship. Records from earlier centuries are frequently incomplete. Documents are lost. Archives are damaged. This is the reality of working with historical evidence from any era before modern record-keeping practices were standardized. But the problem is that the incompleteness is not uniform. In many other regions of the world, historical documentation from the 17th, 18th, and 19th centuries is fragmentary but recoverable. You can trace the outlines of populations, their movements, their cultural practices, and their demographic composition through a combination of surviving documents, archaeological evidence, and genetic data. The picture is incomplete but coherent. In the regions under examination here, the incompleteness is more severe and more specifically concentrated in exactly the time period that would be most relevant to understanding whether a significant population change occurred. This is not universal across all historical periods in these regions. Documentation from earlier periods, the medieval era, for example, often survives in better condition. The gap concentrates in the specific centuries that this investigation keeps returning to. The fourth line of evidence brings the argument back to where it started. Almost all of the blood type data collected from Eastern Europe and Central Asia was gathered after the major disruption periods had already concluded. The Russian Empire's expansion into Central Asia was largely complete by the mid-19th century. The population reorganizations of the 17th and 18th centuries were long finished before the first blood type surveys were conducted. The blood type studies of the 1910s and 1920s were measuring the populations that existed after all of this had happened, not the populations that existed before. This means the data that forms the foundation of the blood type map for these regions was collected from the end state of a process, whatever that process was, rather than from the populations that preceded it. And using end-state measurements to make claims about the pre-process populations requires an assumption of continuity that the other three lines of evidence are actively calling into question. This is where the combination matters. Each of these four observations, taken alone, has a reasonable individual explanation. Ancient DNA anomalies can be attributed to sampling limitations. Geographic overlap between anomalies and disruption zones can be attributed to coincidence or to the fact that disrupted regions naturally produce more complex data. Historical documentation gaps can be attributed to the general limitations of pre-modern record-keeping. And post-disruption blood type measurement can be attributed simply to the timing of when the relevant science was developed. None of these individual explanations is implausible, but applying all four of them simultaneously, invoking a different independent explanation for each of four converging lines of evidence, all pointing toward the same region and the same time period, starts to require a significant amount of analytical effort, and analytical effort applied in the service of preserving an existing model, rather than in the service of following the evidence, is worth examining critically. In scientific methodology, there is a concept sometimes described as the weight of converging evidence. It refers to the cumulative significance of multiple independent lines of evidence that point toward the same conclusion, even when no single line is individually conclusive. The weight of converging evidence is precisely why science rarely requires a single definitive proof in complex historical or biological questions. Instead, it looks at whether multiple independent sources of information align more consistently with one interpretation than with another. In this case, for independent lines of evidence, ancient DNA research, geographic overlap patterns, historical documentation gaps, and measurement timing all align more consistently with the interpretation that something significant happened to the populations of Eastern Europe and Central Asia in the period between 1600 and 1900 than with the interpretation that these populations developed continuously and gradually from ancient times into the modern period. The standard narrative of human population history across Eurasia describes a process of continuous, gradual development, ancient populations evolving slowly over millennia, mixing with neighboring groups along established migration routes, adapting to environmental pressures, and producing the modern populations that blood type surveys eventually measured in the 20th century. This narrative is not wrong in its broad outlines. The broad arc of human prehistory, the out-of-Africa migrations, the spread of agriculture, the major Bronze Age population movements, is well supported by genetics and archaeology, but broad outlines can accommodate significant specific disruptions without those disruptions being visible at the level of the overall narrative. A model that accurately describes the general shape of a process can still miss important details that occurred within that process, and the details that appear to be missing from the standard narrative of Eurasian population history are not small ones. Some researchers have proposed that the standard narrative contains what they describe as an unacknowledged breakpoint, a moment, or more likely a period spanning several generations, in the 17th, 18th, or 19th century, when the population trajectory of certain Eurasian regions was significantly interrupted. Not a minor disruption that the population recovered from quickly and completely. A more substantial break. One that altered the genetic composition of the affected regions in ways that are still detectable, if incompletely, in the blood type and genetic data available today. This breakpoint, if it exists, would explain why ancient DNA studies consistently produce surprising results in these regions. It would explain why the geographic anomaly clusters overlap with the disruption zones. It would explain why the historical record gaps concentrate in exactly the relevant centuries. And it would explain why the blood type measurements collected after the disruption period Do not project cleanly backward onto the ancient DNA record. The breakpoint would be the common underlying cause, the thing that transforms four separate anomalies into a single coherent pattern. Whether such a breakpoint actually exists, and if so, what caused it, whether natural catastrophe, deliberate population replacement, epidemic disease, or some combination of factors, is not something the current evidence can definitively establish. The pattern is visible. The convergence of independent lines of evidence is real, but the specific mechanism behind the pattern remains genuinely unclear. And that is precisely what makes this investigation worth continuing. Because a pattern without a clear explanation is not a closed question. It is an open one. And open questions, when they appear consistently across multiple independent lines of evidence, pointing toward the same unacknowledged region of uncertainty, are usually pointing toward something that has not yet been fully seen. Up to this point, the investigation has focused primarily on blood type data, on the gaps in the measurement record, on the anomalies in ancient DNA studies, on the geographic clustering of unexpected findings, and on the historical disruptions that occurred in the regions showing the strongest discrepancies. Blood type has been the central thread, but blood type is not the only thread, and this is where the problem begins to expand in a way that is difficult to ignore. Because if a large-scale population replacement occurred in Central Eurasia during the 17th, 18th, or 19th century, the evidence of that replacement would not appear only in blood type data. It would appear across multiple independent categories of evidence simultaneously. Language, architecture, genetics beyond blood type, oral history. And when you look at these other categories carefully, you find something unexpected. The same kind of anomaly, the same kind of pattern, the same kind of gap between what the standard model predicts and what the evidence actually shows. Start with language. Language is one of the most powerful tools available for reconstructing the history of human populations. When populations share a common ancestry, their languages tend to share structural similarities, similar grammar patterns, related vocabulary, comparable sound systems. These similarities are not random. They reflect the shared origin of the populations that speak those languages. Linguists have mapped these relationships across thousands of languages worldwide, building detailed family trees that trace the branching and diverging of languages over thousands of years. This field, comparative linguistics, is mature, rigorous, and well-established. And in most parts of the world, the language family trees it produces align reasonably well with the population history reconstructed from genetics and archaeology. The linguistic relationships and the genetic relationships tend to point in the same direction, because both reflect the same underlying history of who came from where and who mixed with whom. But in Central Eurasia, this alignment is less clean. There are languages spoken in this region whose structural relationships to neighboring languages are difficult to explain through the standard model of linguistic development. Languages that should, based on geographic proximity and centuries of documented contact, show clear structural similarities to their neighbors, but don't. The differences are not minor vocabulary variations of the kind that develop naturally between related languages over time. In some cases, they are deep structural differences. Differences in how sentences are constructed, how grammatical relationships are marked, how the sound system is organized. These kinds of deep structural differences typically indicate either very long separation between populations, more than the geographic proximity would suggest, or a more recent break in continuity, a point at which one population was replaced by another that spoke a fundamentally different kind of language. Some linguists working on Eurasian language distributions have proposed population replacement as the most straightforward explanation for certain puzzles in the linguistic record. This is not a fringe position. Population replacement as a driver of linguistic change is well documented in mainstream linguistics. The replacement of indigenous languages by colonial languages in the Americas, in Australia, and in parts of Africa is a clear modern example of how rapidly and completely language distributions can change when one population displaces another. The argument some researchers are making is that similar replacement events may have occurred in Central Eurasia at earlier periods, events that altered the linguistic landscape of the region in ways that the standard model of gradual linguistic drift cannot fully account for. Whether those events fall within the time period this investigation keeps returning to, the 17th through 19th centuries, is not established clearly, but the structural anomalies in the linguistic record are real, and they raise questions that do not yet have fully satisfying answers. Architecture and material culture present a different category of evidence, but one that points in a related direction. Across certain parts of Central Asia and Eastern Europe, there are architectural structures whose origins are debated, whose construction methods are not clearly attributed to known historical populations, and whose age is in some cases genuinely uncertain. This is not as unusual as it might sound. Attribution disputes in archaeology are common, and the difficulty of definitively dating some forms of construction means that genuine uncertainty about who built what is a regular feature of the field. But what some researchers have noticed is that these attribution disputes and construction mysteries cluster, again, in the same regions and the same general time periods that produce the other anomalies this investigation has been tracking. That geographic and temporal clustering is becoming a recurring feature of this analysis, and recurring patterns in independent categories of evidence are the kind of signal that serious investigation needs to take seriously. Now move to genetic haplogroups. This is where the connection to the blood type question becomes most direct and most significant. Haplogroups are genetic markers inherited through specific lines, either through the father, via the Y chromosome, or through the mother, via mitochondrial DNA. Unlike blood type, which is determined by a combination of genes from both parents, haplogroup markers are passed intact from one parent through successive generations, without recombination. This makes them extraordinarily stable over time. A haplogroup present in a population 10,000 years ago will still be identifiable in the descendants of that population today, largely unchanged. This stability is precisely what makes haplogroups so valuable for tracing ancient population movements. They are genetic labels that persist across millennia, allowing researchers to follow population lineages through time in a way that other genetic markers cannot. The distribution of haplogroups across Central Asia and Eastern Europe contains patterns that the standard model of population history struggles to accommodate comfortably. In several regions, haplogroup frequencies show abrupt transitions, points in the geographic distribution where one haplogroup cluster ends and a different one begins, without the gradual blending that would be expected if the transition reflected slow, continuous migration and mixing. Abrupt haplogroup transitions are a recognized signature of population replacement in population genetics. They appear in the archaeological record of Europe, for example, where the transition from hunter-gatherer populations to farming populations is marked by exactly this kind of abrupt haplogroup shift. Researchers have documented this transition thoroughly and attribute it to a relatively rapid replacement of one population by another carrying a different haplogroup signature. The same interpretive framework, abrupt transition as evidence of replacement, is applicable to the pattern seen in Central Asian and Eastern European haplogroup distributions. But applying it there leads to conclusions that the standard historical narrative has not fully integrated. The connection between haplogroup patterns and blood type patterns is not indirect. It is direct and straightforward. Both blood type and haplogroup are genetic traits. Both are inherited and stable over time. Both reflect the underlying genetic composition of the population carrying them. If a population replacement occurred, if a group of people with one genetic profile was substantially displaced by a group with a different genetic profile, both the blood type distribution and the haplogroup distribution of the affected region would shift simultaneously. Not one without the other. Both. This is important because it means the blood type anomalies and the haplogroup anomalies are not independent puzzles. They may be two visible expressions of the same underlying event, and an event that produces simultaneously detectable signals in two independent genetic marker systems is a more significant event than one that affects only one marker in isolation. This is where it becomes unclear in a way that the standard framework has not fully addressed. If the haplogroup patterns in certain regions suggest population replacement, and if the blood type patterns in the same regions show anomalies consistent with population replacement, and if the ancient DNA studies in the same regions produce distributions that don't match the modern populations, then these three independent genetic signals are all pointing toward the same conclusion about the same region. The standard response is to invoke separate explanations for each signal: ancient migration for the haplogroups, sampling error for the blood type anomalies, methodological limitations for the ancient DNA discrepancies. But as this investigation has noted before, invoking a separate independent explanation for each of several converging signals is a progressively less economical analytical strategy. At some point, the simpler explanation is that the signals are converging because they share a common cause. Oral histories add another dimension to this picture, though they are the most difficult category of evidence to assess rigorously. In certain communities across Central Asia, oral traditions have preserved accounts of population disruptions, displacement events, and cultural discontinuities that mainstream historical scholarship has not fully engaged with. Oral history is not a reliable source for precise historical detail. It transforms over generations, incorporates mythological elements, and reflects the concerns of later storytellers as much as the realities of past events. These limitations are well recognized by researchers who work with oral traditions. But oral history can preserve the general shape of significant historical events even when the specific details have been transformed by generations of retelling. The consistent presence across multiple independent oral traditions in Central Asian communities of accounts describing major disruption and displacement events in the relatively recent past is worth noting, not as direct evidence, but as a corroborating signal that adds to the pattern. What is emerging from this expanded view is a picture that is considerably more complex than the standard population history of Central Eurasia presents. The standard picture describes a region with ancient roots, documented migration events, and a relatively continuous demographic development from prehistory into the modern period, shaped by the rise and fall of nomadic empires, but fundamentally continuous in its human composition. That picture is not obviously wrong at the level of broad outline, but the specific evidence, genetic, linguistic, architectural, and oral, contains enough anomalies, enough clustering of unexpected findings, and enough convergence of independent signals to suggest that the specific details of that picture may be significantly incomplete. And this is where the explanation becomes difficult, because accounting for all of these anomalies within the standard framework requires either accepting a very large number of separate, unrelated explanations for individually unrelated problems, or accepting that the standard framework itself is missing something that occurred in a specific region, during a specific period, at a scale significant enough to leave detectable traces across multiple independent categories of evidence. What that something was: natural disaster, epidemic, deliberate replacement, poorly documented conquest, or some combination of factors, is not specified by the evidence available. The evidence points toward the existence of a significant unacknowledged event. It does not, by itself, identify the nature of that event. The picture, as this investigation has now established through multiple lines of independent evidence, does not become clearer the more carefully you examine it. It becomes less clear. Details that seemed well explained at a surface level reveal complications when examined closely. Patterns that the standard model accommodates at a broad scale show specific inconsistencies at a finer scale. And the inconsistencies, rather than appearing randomly distributed, consistently cluster in the same geographic zones and the same historical periods. That is the kind of pattern that an investigation cannot simply note and move past. It is the kind of pattern that demands a better explanation than has so far been offered. And whether that better explanation eventually confirms, modifies, or entirely replaces the current standard account of Central Eurasian population history is a question that the available evidence has not yet answered. At this point in the investigation, a reasonable question emerges naturally. If the blood type anomalies are real, if the haplogroup patterns are genuinely unusual, if the linguistic and historical gaps are as significant as the evidence suggests, then what do researchers actually think is behind all of this? The answer is not simple, and it is not unified. Different researchers, approaching the same body of evidence from different starting assumptions and different methodological traditions, have arrived at very different interpretations. These interpretations do not all point in the same direction. Some are compatible with each other, others are not. And the fact that serious people looking at the same data can reach such different conclusions is itself an important part of the picture. It tells us something about how unresolved the underlying questions actually are. The first interpretation comes from researchers working largely outside mainstream academic institutions. This group argues that the blood type anomalies and the genetic irregularities are not accidental. They are not the product of natural migration events or random demographic fluctuation. Instead, these researchers propose that the anomalies are consistent with a deliberate and organized replacement of a specific population, a population that carried distinct biological markers, including specific blood type distributions that differed significantly from the populations that came after. This interpretation is closely associated with the broader framework, sometimes called the Tartarian reset hypothesis. In its most developed form, this hypothesis proposes that a large and technologically sophisticated civilization once occupied Central Eurasia, that this civilization was not simply displaced by conquest or gradually absorbed through migration, that it was systematically removed from history, its people replaced, its records destroyed or rewritten, and its physical and cultural legacy attributed to other historical actors or simply erased from the official account. This is a significant claim, and it is important to be clear about what kind of evidence is offered in support of it, and what the limitations of that evidence are. The researchers working within this framework point to the cartographic evidence, the consistent labeling of Grand Tartary on pre-18th century European maps. They point to what they describe as architectural anomalies in the region, structures whose design and construction, they argue, cannot be cleanly attributed to the known historical populations. They point to the documentary gaps in the historical record, and they point to the blood type and genetic anomalies that this investigation has been tracking. Each of these pieces of evidence is real. The maps existed. The documentary gaps are genuine. The genetic anomalies are published in peer-reviewed research. But the interpretation placed on this evidence, that it reflects a deliberate, organized erasure of an advanced civilization, goes significantly beyond what the evidence itself can demonstrate. The evidence shows that something unusual may have happened. It does not show that what happened was deliberate, coordinated, or involved a specifically advanced civilization. That additional layer of interpretation requires a degree of certainty the data does not provide. The second interpretation is considerably more cautious, and it comes from researchers who do not reject the anomalies, but who resist the conclusion that anything unusual or deliberate is responsible for them. This group acknowledges that ancient DNA studies have produced surprising results. They acknowledge that haplogroup distributions in certain regions are difficult to explain with perfect precision, and they acknowledge that the historical record for Central Eurasia contains gaps that are more severe than what you see in many other regions. But their explanation for all of this is different. They argue that the anomalies reflect poorly understood ancient migration events, movements of people that occurred thousands of years ago, and that left genetic traces, which current models have not yet fully integrated. In this view, the problem is not that something unusual happened in the 17th or 18th century. The problem is that the ancient history of Central Eurasia is genuinely complex, and the models used to interpret it are not yet sophisticated enough to capture that complexity without producing apparent anomalies. This is a scientifically respectable position. Population genetics is a young field relative to the timescales it studies. The models it uses are continuously being revised as new data becomes available. What looks like an anomaly today may have a straightforward explanation that emerges from future research. The history of science contains many examples of apparent anomalies that turned out to be artifacts of incomplete models rather than evidence of genuinely unusual events. And in a field as complex as Eurasian population history, involving thousands of years of migration, mixing, adaptation, and disruption across an enormous and climatically diverse territory, incomplete models are not only possible but expected. The gaps in understanding, in this view, reflect the limits of current science, not the existence of an unacknowledged historical event. But this is where the second interpretation runs into its own difficulties. The argument that ancient migration complexity explains the anomalies is plausible in general terms, but it becomes less satisfying when applied to specific cases. The geographic clustering of the anomalies, their consistent concentration in the same regions that experienced the most severe historical disruptions in the 17th through 19th centuries, is difficult to explain through ancient migration events that predate those disruptions by thousands of years. If the anomalies were caused by poorly understood ancient migrations, you would expect them to be distributed more broadly, reflecting the broad distribution of ancient population movements across Eurasia. The fact that they concentrate specifically in regions with severe recent disruption histories suggests a more proximate cause than ancient complexity alone can explain. The third interpretation comes from within mainstream population genetics, and it targets the reliability of the evidence itself rather than offering an alternative account of what that evidence reflects. Researchers in this camp argue that the anomalies in blood type data, particularly the early 20th century survey data, and its inconsistencies with genetic projection models, are largely artifacts of methodological limitations in the original studies. The early blood type surveys conducted in the 1910s and 1920s were, by modern standards, working with small sample sizes. Sampling bias, the tendency for samples to overrepresent accessible or cooperative population groups, was a known problem that early researchers did not always have the tools to adequately control, and the geographic coverage of these early studies was uneven, with some regions studied more thoroughly than others. These are legitimate methodological concerns. Small sample sizes produce unreliable frequency estimates. Sampling bias can distort distributions in ways that persist through subsequent analyses if the biased data becomes the baseline for later work. And uneven geographic coverage means that the global blood type map, in its early form, was more of an approximation than a precise measurement. If the anomalies in the data are artifacts of these methodological limitations, then they do not reflect real population differences at all. They reflect the noise inherent in early, imprecise measurement, and as methods improve and sample sizes grow, the anomaly should gradually disappear, not because something new has been discovered, but because the cleaner data reveals that the apparent inconsistencies were measurement errors all along. At first, this explanation seems compelling, and in some specific cases, it probably is correct. Some of the anomalies in early blood type data almost certainly do reflect sampling limitations rather than real population differences. But the explanation becomes harder to sustain when applied to the full body of evidence this investigation has reviewed. Ancient DNA studies use entirely different methods from early blood type surveys. They are not subject to the same sampling biases. They do not rely on the same early 20th century data, and yet they produce anomalies that overlap geographically with the anomalies in the blood type data. If the blood type anomalies were purely methodological artifacts, you would not expect ancient DNA research, using completely different techniques on completely different material, to produce overlapping patterns of unexpected findings in the same regions. The overlap between two independent methodologies suggesting the same regional anomalies is difficult to explain as coincidental measurement error in both systems simultaneously. The fourth interpretation focuses on epidemic disease, and of all the alternative views presented here, this one operates most comfortably within the mainstream scientific framework, while still acknowledging that something significant happened to alter the genetic composition of certain populations. The argument is straightforward. Central Eurasia was repeatedly devastated by epidemic disease between the 14th and 19th centuries. The Black Death of the 14th century is the most famous example, but it was not the only one. Subsequent waves of plague, combined with smallpox, cholera, and other epidemic diseases, moved through these populations over 5 centuries of repeated exposure. Each epidemic wave had the potential to create a population bottleneck, dramatically reducing the number of survivors and thereby altering the genetic composition of the surviving group. Blood type frequencies can shift significantly through bottleneck events because the survivors are not a representative sample of the pre-epidemic population. This explanation has the considerable advantage of invoking well-documented historical events. The epidemics are real. Their demographic impact is acknowledged in mainstream historical scholarship. The mechanism by which epidemic bottlenecks alter blood type frequencies is well understood in population genetics, and the geographic distribution of these epidemic events, heavily affecting Central Asia and Eastern Europe, overlaps with the regions showing the strongest anomalies in the blood type and genetic data. This is not a speculative connection. It is a well-established relationship between known historical events and a known biological mechanism. But this is where even the epidemic explanation shows its limits. Epidemic bottlenecks alter blood type frequencies by creating a non-representative surviving sample from an existing population. The survivors carry a subset of the original genetic diversity. This produces measurable shifts in blood type frequency within the surviving population and its descendants. But it does not produce the same kind of genetic signal as population replacement. When a bottleneck occurs, the genetic markers of the original population are still present in the survivors, just in different proportions. The haplogroup distribution remains recognizable as descended from the original population, even if some lineages are lost. Ancient DNA from the pre-bottleneck period should still show clear continuity with the post-bottleneck population, just with somewhat different frequencies. If what researchers are finding is something that looks less like altered frequencies in a continuous population and more like a fundamental discontinuity, one population ending and a different population beginning, then epidemic bottleneck alone is not sufficient to explain the pattern. And some of the ancient DNA findings in the regions under examination look more like the second type of pattern than the first. What emerges from examining these four interpretations together is a landscape of genuine uncertainty. Each view captures something real. The deliberate replacement hypothesis points to a real pattern of converging anomalies, even if its specific claims go beyond what the evidence supports. The ancient complexity view correctly identifies that Eurasian population history is genuinely difficult to model with current tools. The methodological critique correctly identifies real limitations in early blood type data, and the epidemic hypothesis correctly identifies a powerful and documented mechanism for demographic change in exactly the right time period and geographic region. The problem is that none of them, individually, accounts for the full body of evidence that this investigation has assembled. Each resolves some of the anomalies, while leaving others unexplained. Each fits some of the data while struggling with other parts of it, and the areas where each interpretation struggles tend to be the areas where the pattern of converging evidence is strongest and most difficult to dismiss. This does not mean the truth lies in any one of these four positions. It may lie in a combination of them. Epidemic disease reducing a population, followed by partial replacement through migration, occurring in a region whose historical record was subsequently managed by the incoming administrative power. That kind of multi-factor account might explain more of the evidence than any single interpretation can. But even that more complex account would need to be tested rigorously against the full body of genetic, linguistic, archaeological, and historical evidence before it could be accepted with confidence. And that kind of rigorous, comprehensive testing has not yet been done. The field is still in the stage of identifying the anomalies and debating what category of explanation they belong to. The specific mechanism remains, at this point in the investigation, genuinely open. And that openness, that space between the evidence and a satisfying explanation, is where the most important questions in this story continue to live. There is a particular kind of discomfort that comes at the end of a genuine investigation, not the discomfort of having found something disturbing, though that can be part of it, but the discomfort of having followed a thread carefully, methodically, and honestly, and arriving not at a clear answer, but at a clearer picture of how much remains unknown. That is where this investigation now stands. Not at a resolution, not at a point where the evidence assembles neatly into a single, confident explanation, but at a place where the questions themselves have become sharper, more specific, and more difficult to dismiss than they were at the beginning. And that sharpening of the questions is itself a form of progress, even if it does not feel like one. Begin with the most basic question this investigation raised at the outset. Why does large-scale blood type data only exist from the 20th century onward? The timing seems, on the surface, like a simple matter of scientific history. Blood types were not discovered until 1901. Large-scale population surveys require infrastructure, resources, and standardized methodology that only became available in the early decades of the 20th century. So the data begins when the capacity to collect it begins. That is a reasonable explanation, and in the broadest sense, it is accurate. But the investigation has shown that the timing is not merely a neutral feature of scientific development. It is also a feature that has direct implications for what the data can and cannot tell us, because the period immediately before the data begins, roughly 1600 to 1900, is precisely the period during which the most significant population disruptions in the affected regions occurred. The absence of data is not evidence that nothing changed. It is a window of invisibility that covers exactly the time when change, if it occurred, would have been most visible. This is the first unresolved question, and it is unresolved not because no one has thought about it, but because the information needed to answer it does not exist in a recoverable form. You cannot measure what was not measured. You cannot reconstruct with precision a blood type distribution that no one ever recorded. Ancient DNA research is the closest available tool for looking backward into this invisible window. But as this investigation has shown, ancient DNA research in the relevant regions consistently produces results that complicate rather than confirm the standard model. The unexpected distributions, the abrupt transitions, the points in the archaeological record where one genetic profile ends and a different one begins without the gradual blending that continuous population development would produce. These findings do not answer the first question. They deepen it. They suggest that something worth measuring was happening in that invisible window, and that whatever was happening left traces in the biological record that the current framework is not fully equipped to interpret. The second unresolved question follows directly from the first. Why do ancient DNA studies consistently produce blood type distributions that don't match what the standard model of gradual, continuous population development would predict? The individual cases have individual explanations. Migration events, sampling limitations, regional isolation producing unusual local frequencies. These explanations are available and have been applied, but the consistency of the unexpected findings, the fact that the surprises cluster in specific regions and specific time periods rather than appearing randomly, resists being fully absorbed by case-by-case explanations. When a pattern appears consistently across independent studies, conducted by different research teams using different methodological approaches, the pattern itself becomes a data point. And the data point here is that the standard model consistently struggles to account for what ancient DNA research finds in Central Eurasia and Eastern Europe. That struggle is not a minor footnote. It is a structural feature of the current state of knowledge in this field. Some researchers argue that the struggle reflects the genuine complexity of ancient population movements, migrations and admixture events that occurred thousands of years ago, and that current models have not yet captured with sufficient precision. Others argue that the struggle reflects a more proximate cause, events in the historical past few centuries that the models are not accounting for because those events are not well represented in the standard historical narrative. Both positions acknowledge the struggle. They disagree about its source, and that disagreement itself is unresolved. The tools to definitively distinguish between ancient complexity and recent replacement are not yet available in a form that can be applied comprehensively to the relevant regions with the sample sizes and geographic coverage needed to produce definitive results. This is not a failure of individual researchers. It is a genuine frontier of current knowledge. The third unresolved question is perhaps the most structurally interesting. Why does the geographic overlap between blood type anomalies and the historical territory of Tartaria appear repeatedly across independent lines of evidence? This question is significant because the overlap is not the product of a single analytical framework. It does not appear only in blood type data. It does not appear only in haplogroup distributions. It does not appear only in ancient DNA studies. It appears across all of these independently, and it also appears in the linguistic anomalies, in the architectural attribution disputes, in the historical documentation gaps, and in the cartographic record of a geographic designation that dominated European maps for generations before disappearing with unusual speed and completeness. Each of these independent lines of evidence was developed using different methods by different researchers, asking different questions, and yet they all return, repeatedly, to the same geographic zone. At first, this might seem like confirmation bias, the tendency to see patterns that align with a pre-existing framework, and that possibility cannot be entirely dismissed. If researchers are looking for anomalies in a specific region because they expect to find them, they may find them more readily than a neutral analysis would produce. This is a legitimate methodological concern, and it is one that any honest assessment of this body of evidence must take seriously. But confirmation bias does not easily account for anomalies that appear in peer-reviewed ancient DNA research, in mainstream linguistic scholarship, and in the published analyses of population geneticists who were not specifically looking for evidence related to Tartaria or any alternative historical hypothesis. When anomalies appear in research that was not designed to find them, research conducted by people with no particular investment in alternative historical frameworks, the confirmation bias explanation becomes less efficient. The fourth unresolved question concerns the historical record itself. Why does the documentation for Central Eurasia during the critical 17th through 19th century period contain more gaps, contradictions, and missing material than comparably sized regions in other parts of the world? Mainstream scholarship offers several responses to this question. The regions in question were large and sparsely populated in some areas, making comprehensive record-keeping difficult. The administrative transitions of the period, the expansion of the Russian Empire, the fragmentation of Central Asian political structures, created conditions in which existing records might not survive. Fires, floods, wars, and simple neglect account for the loss of enormous amounts of historical documentation in many parts of the world. None of these explanations is implausible, but this is where the explanation becomes difficult, because the documentation gaps are not uniformly distributed across the period or across the region. They concentrate in specific time periods and in specific types of documents, precisely the kinds of records that would be most informative about the demographic composition of the affected populations. Internal administrative records, census information, religious and community documentation that would normally accumulate within a functioning society. These are the records that are most systematically absent, and their absence is most acute during the period that this investigation keeps returning to. The pattern of absence is not random. Random documentary loss produces gaps scattered across different types of records and different time periods. Systematic gaps concentrated in specific document types during specific periods are a different kind of pattern, and a different kind of pattern suggests a different kind of cause, though what that cause specifically was remains, at this point, genuinely unclear. The official explanation for the state of the evidence, the framework that mainstream science and mainstream history provide, covers the broad outlines of this story adequately. Blood types evolved over millions of years. Populations migrated and mixed over thousands of years. The historical disruptions of the 17th through 19th centuries reshaped demographic distributions in ways that are still being fully understood. The gaps in the record reflect the normal limitations of historical science applied to a complex and turbulent period. This framework is coherent. It is supported by substantial evidence at the level of broad principle, and it provides a working account of human population history across Eurasia that is useful, even if incomplete. But the specific details, the places where the broad framework meets the fine-grained evidence, are where the difficulties appear. The specific blood type anomalies that the broad framework cannot fully absorb, the specific haplogroup transitions that look less like gradual drift and more like abrupt replacement, the specific linguistic puzzles that population replacement explains more efficiently than gradual drift does, the specific cartographic record of a geographic designation that disappeared with a speed and completeness that the standard account of imperial expansion does not entirely account for. At the level of specifics, the official framework works harder than a well-confirmed model should need to work, and the places where it works hardest are consistently the same places, the same region, the same time period, the same categories of evidence. The alternative theories, the Tartaria reset hypothesis, and its associated frameworks, point toward these specific difficulties accurately. They correctly identify that something in the standard account is not fully resolved, but they move from that correct identification to specific claims that the evidence does not yet support. The claim that the replacement was deliberate, the claim that the replaced population was technologically advanced, the claim that the erasure was coordinated across multiple competing powers. These specific claims require a level of evidential support that the available data does not provide. The alternative frameworks are better at identifying the questions than at answering them, and a framework that asks the right questions but provides unverified answers is a starting point for investigation, not a conclusion. What seems reasonably clear, after following this evidence through 9 chapters of careful examination, is that the biological history of human populations across Eurasia in the 17th 18th, and 19th centuries is not as well understood as standard textbooks suggest. The textbook picture of continuous, gradual population development from ancient times into the modern period, shaped by well-documented migration events and recorded historical processes, is a simplification. It may be a useful simplification. Simplifications often are. But a simplification that cannot account for the specific anomalies visible in ancient DNA data, haplogroup distributions, blood type surveys, linguistic patterns, and historical documentation is a simplification with a significant gap at its center. And that gap is located precisely where this investigation has been looking, in the specific populations, the specific time periods, and the specific geographic regions that the evidence keeps returning to. The data that exists does not tell a single clean story. It tells several partially overlapping stories, each supported by some of the evidence and contradicted by other parts of it. The picture those fragments form depends significantly on which pieces you examine first, which framework you bring to the analysis, and which anomalies you treat as central versus peripheral. A researcher beginning with the assumption of continuity will find much in the data to support that assumption, while explaining the anomalies as peripheral noise. A researcher beginning with the assumption of disruption will find much in the data to support that assumption, while treating the apparent continuities as artifacts of incomplete measurement. Neither starting point is obviously wrong, and neither produces a picture that the other cannot challenge with legitimate counter-evidence. This is not a comfortable place to end an investigation. Comfortable endings involve resolution. They involve the evidence pointing clearly in one direction and the conclusion following naturally from it. This investigation does not end that way. It ends with the questions sharper than they were at the beginning, and the answers no more certain. The blood type data raises questions about what existed before it was measured. The ancient DNA findings raise questions about whether the populations that were measured are genuinely continuous with the populations that preceded them. The historical record raises questions about why the documentation is thinnest precisely where the investigation needs it most. And the geographic overlap between all of these anomalies raises the broadest question of all, whether something significant happened in a specific region during a specific period that the current standard account of human history has not yet fully seen, fully measured, or fully explained. That question remains open, and open questions, followed honestly and carefully, are where genuine understanding eventually begins.